Hello, I'm Peter Brooks from University Sunshine Coast Honey Research Lab, and I'm going to present today on wound healing Australian Manuka honeys. First of all, I'd like to thank Bridget for inviting us along to the Apitherapy Association conference, and also to all the students that contributed to this project. So I'll start sharing my screen. Okay, first of all, what is Manuka honey and how does it help clear wounds? All right, let me just Well, Manuka honey is, we consider to be any honey produced by the honeybee from the Leptospermum genus. Now, New Zealand has one species of Leptospermum scoparum, and Australia has 84 species, including the scoparum species. Now, before I get into the politics honey or avoid the politics honey, we're going to assume that all Leptospermum species from Australia and New Zealand are equally called Manuka honey because it's the active ingredients within those that give them the beneficial properties of a Manuka honey and its wound healing and antibacterial properties. Some of the Australian species are more active than New Zealand, some aren't, but also depends on how much leptosperm nectar the bees actually bring in. They can forage for up to 3 to 5 kilometers from the hive, so they'll naturally blend leptosperm nectar with other nectars from other species. So you can have a very high Manuka activity or a very low Manuka activity from both countries. It doesn't matter where it's from, it's the active ingredients within the honey that give it this bioactivity that everybody's interested in. So why so interested in honey as a wound treatment? Well, before the advent of antibiotics, they were widely used, but went out of fashion when antibiotics were discovered. Now, with the rise of the superbugs, it's turning out that there's a renewed interest in them because superbugs, the golden staffs and so on, are now taking over wounds, becoming multi-resistant, have not been able to develop any resistance to honey's multiple treatments. So there's a renewed interest both for the antibacterial action because the bugs aren't overcoming it, and also for the wound healing properties overall of the honeys. So let's just jump into some of the activity of honeys. All honeys have a certain activity of antibacterial action. They're all hygroscopic, where they draw water out of the wound and also desiccate the bacteria. They all are slightly acidic, and bacteria don't like the acidic environment. And many honeys produce hydrogen peroxide on dilution. So as they're drawing out the extrudants from the wound, if applied to a wound, for example, they become diluted and they start producing hydrogen peroxide, natural antibacterial action. But these leptosperms, or Manukas, whichever way you like to call it, have a special activity that's derived from the plant. They're even there when the peroxide's knocked out. So that's known as non-peroxide activity. So connecting the chemistry to the biology of these honeys, we're relating how much this antibacterial action and non-peroxide activity is there due to the natural antibacterial means that's within the honeys. So let's just talk about Manuka honeys for a sec. They draw the infection out of the wound and break up the biofilms that are protecting the pathogens. Many pharmaceuticals can't do that. And then once they've drawn that infection out of the wound, broke up the biofilm, they're now able to kill the bacteriums. So kill the infections. They also dampen down the inflammation and promote wound tissue regeneration, sorry. My apologies for the photos on this slide, but these are of an old lady's ulcerated, badly infected leg that was not responding to modern medications, but responded, as you see, quite nicely in Manuka honeys. So I'm going to circle back this wound healing property of anti-inflammation activity of those honeys later on as well. Let me just run through a bit more on the chemistry of these things. This non-peroxide activity was discovered when golden staff plates were tested. Let me run through what you're looking at the moment. These are agar plate with golden staff growing on it. The upper row up here contains the antiseptic phenol in various concentrations, forming a standard curve. You might say, The circle or the zone of inhibition is where the bacterium has been knocked out by the phenolic standards. Down the bottom here, some honeys. These first four are Manuka honeys, and this one here over here is clover. They've had catalase added to them, so they knock out the peroxide activity. So any antibacterial action is now due to non-peroxide action. See, the clover has no zone of inhibition around it, so it has zero non-peroxide activity. But these other Manuka honeys do still retain the zone of inhibition after hydrogen peroxide has been knocked out. So this is referred to as the non-peroxide activity, which leads to market value of the honeys, also leads to better patient outcomes when applied to wounds, obviously. So this methyl glycoside is responsible for that, and I'll show you how we discovered that, or found that in Australian leptospermum honeys shortly. Is measured in ppm or parts per million. You'll see this written on jars of honey. If you go and purchase one of these Manuka honeys, you see MGO rating. Now, it's also paid to the beekeepers on that MGO rating. So our lab actually set up the method. They actually measure the methyl glycoside, which is responsible for this non-peroxide activity. The higher the non-peroxide activity, the higher the MGO rating of that honey. But MGO, this methyl glycoside compound here, is 3-carbon dicarbonyl. It's not present in the nectar of the Manuka plant, it's actually in the honey. Instead, there's a 3-carbon sugar called dihydroxyacetone that's present. And this dihydroxyacetone here starts off in the nectar, and as the honey matures over 12, 24 months in the honey, develops in methyl glycoside. So a young honey that's come from Manuka will have high dihydroxyacetone, low MGO, and over a period of 12 to 24 months, when treated properly, that methyl glycoside will increase and the honey will become more and more medically active antibacterial honey. And the study we did, culminating in 2016, showed that didn't matter which of the leptosperm it came from, the methyl glycoside was very much correlated to this methyl glycoside activity. And I'll show you some calibrations later on. It shows how the methyl glycoside comes from. So these are some bees foraging on leptosperm species, collecting that nectar, bringing it back, making honey, and therefore having this activity that's now associated with Manuka honey. So in our test, we looked at the dihydroxyacetone nectar slowly over 1 to 2 years forming methyl glycoside. We also looked at this age marker HMF. This HMF age and heat marker increase if your honey goes very old and dark and caramelized, or if you try to heat it to try to accelerate this reaction very quickly. We showed that's not a good thing to happen because it actually destroys the methyl glycoside as well. But let's see rocking on about these bioactive compounds that are present in the honey. Our analysis separated out dihydroxyacetone, the HMF, and methyl glycoside. So this particular honey be approaching its optimum. There's not much DHA left. There's a lot of MGO conversion. By the way, anisole is an internal standard, so everything's measured to our internal standard of anisole. So we've got something to measure against. And these are the sugars that, 80 to 85% of honeys, all honeys are sugars. So there's a small amount of these materials present in the honey, but they give it that antibacterial action that we're now familiar with. And what we found was that in Australian leptosperm or Manuka honeys, whichever way I like to call it, the non-peroxide activity squared correlated very well with methyl glycoside content. So NPA squared of 100 means the non-peroxide activity was about 10. You notice that that correlates with the antibacterial action beginning. So under 200 or 250 ppm or methyl of this methyl glycoside, or under an NPA of 10, because this is a square up here, we found that it overcomes the activity of the honey. So if you're going to use it for wound dressing, wound healing, you don't want bacteria to overcome it. So this is like a minimum inhibitory concentration to have that level of methyl glycoside in the honey. Now back to the old lady's unfortunate leg that was ulcerated, we found that honeys, or I didn't, but it's been found that these honeys also clear up the wounds quite nicely. We had a friend who had a car accident about 20 years ago. He's been in a wheelchair ever since, and he had pressure sores on his backside that did not respond to normal pharmaceutical creams. But some Australian Manukas did. We gave him some honey, he put it on his backside, and he reported that it cleared up those pressure wounds very nicely. So that's an inflamed wound and not necessarily an infected wound. So we speculated that it wasn't necessarily methylglycol in this case because these Manuka honeys have a very high phenolic content. Phenols are known to be antioxidant. We thought, well, it's a phenol content that's causing this anti-inflammatory action, as seen by some of these publications here. So what one of my students, Danny Melancelli, did, he extracted out the phenolics from various Manuka species and then examined their action in some preclinical trials using cells and cell-free assays. One of the cell-free assays Denny used was against the 5-LOX enzyme. This cascade of reactions down here is caused by a reaction, inflammation of a wound. The wound, ulcerated wounds, sorry, could become inflamed, and if they stick in that flame, you have an ulcerated example of a wound. So what we're hoping to do is show that the phenolics, and this one is methylthreongate, for example, pulled out of the honey matrix, would inhibit the 5-LOX assay, which then would have therefore prevent downstream leukotriene being formed and no more cytokine release, no more reactive oxygen species being released if we can show that phenolics were working in this assay. And lo and behold, they did. Now this is a Scoparum honey from New Zealand, Scoparum honey from Tasmania. You see they follow the same sort of curve, follow the same kind of activity as well. So almost exactly the same activity for both honeys, didn't matter where they came from, either New Zealand or Australia, they had the same activity. So that shows that, at least in the cell-free assay, the work. What about a cell-based assay where we're looking at neutrophils this time and looking to show that honeys were able to dampen down the release of these leukotriene before. So in measuring these cell assays, looking at leukotriene release, we show that both honeys were able to, both phenolics from the honeys were able to prevent the release of leukotriene 4. So they inhibited their release, so showing that another mechanism for which they may actually work. So finally, thank you for listening, and also thank you to those students that helped out during the project. 3 of them have come on to be doctors in their own right. So to thank Amanda Norton, Matt Papalato, Denny Melancelli, and Simon Williams. And also thank you for your attention during this particular presentation. So I'd like to stop now and just say, sorry for my little stumblings. I have Parkinson's, by the way, but thank you for your attention. Bye.